Hello everyone, welcome to the NN Classroom. Today, I'd like to share with you a multiple choice question number 24, extracted from the Specimen paper 2016, that concerning the topic 6, human physiology, our neurotransmission system or our central nervous system. Let's see. The diagram shows the results obtained with an oscilloscope attached to a neuron, so which means we are now understanding the changes in terms of the membrane potential from time to time. Here from X to Y to Z. First of all, students should have recognized that even if they don't tell you, that you should notice, right? The region of X is what we call the region of resting potential. Resting potential is the potential maintained within the membrane, which is going to be roughly about negative 70 millivolt because of the unbalanced distribution of the positive ions, right, into and outside the neuron. So that is what we call the resting potential. So maintaining almost like negative 70, right? And the way from X to Y is a process what we call depolarization, which is a process in which the polarization of the negative 70 disappear or just remove it. So that's a delete the polarization. So from negative 70 all the way normally to the peak of positive 30 millivolt. So that is the point where we have the depolarization. And after the depolarization, we will reconstruct, you see, returning, right? Return. So that's what we call it. Simply means R-E-P-O-L-A-R-I-Z-A-T-O-N. So we reconstruct the polarization from Y to Z. So that is like we reconstruct the polarity. So that's why the normal process of the description of this peak is what we call the action potential, consisting the two processes, depolarization and the repolarization. And sometimes after the repolarization, we will go even lower than the original. So that is a process, but it doesn't tell in this case. But if you see, this is a part, we call it the hyperpolarization. There is some biological significance for having a hyperpolarization that your teacher should have told you. Yeah, but here we will first introduce it first. This is going to be a hyper part. Easy to understand, right? Because you are now doing something more than enough. Enough means the original negative 70 as usual, and you now do it even lower, normally to an area like negative 90 millivolt in terms of the hyperpolarization. So that's why in this case, if you understand the idea behind this oscilloscope trace, then you can realize that for the question asking that why is the change in the oscilloscope occurring between X and Y? What's between X and Y? Especially what I'm now introducing. The point number one, depolarization, right? So depolarization is the process where we remove, so delete the polarity of negative 70 to a positive polarity. So that's why the answer for this question will be so easily goes to D, right? So if you understand that, so you know all the process and all the pathways and in terms of the names from the topic 6.